This is Muredach Reilly here from the University of Pennsylvania. I'm chair of the ATVB Spring Conference, and I'd like to provide some updates on the scientific content of our Spring Conference in Toronto in 2014. This meeting has been a great success with a high attendance, and there's been some great science presented. You know, overall we have some major plenary, 5 plenary sessions and 16 concurrent sessions, as well as our invited awardee speakers and our award-winning speakers. So across that spectrum, we've had some tremendous science, and I just want to sample a little bit of that just to give you some examples. So first, I think that the plenary sessions were particularly good, but I'd like to emphasize perhaps one of my favorite was on the genomics, where we had 3 really strong speakers on Friday morning, and this really encompassed the whole range of modern genomics as it relates to ATVB and peripheral vascular disease themes. This was run in collaboration with our functional genomics council colleagues, and our first speaker was Mike Snyder, who really gave an overview of the application of modern personal omics, from genomics, transcriptomics, to metabolomics, and metabolomics and proteomics, and how to integrate this over time within a person, within an individual, to understand their health and the interaction of the genome with the environment in predicting health outcomes. And this is obviously a controversial and cutting-edge area, but Mike, based at Stanford, is really the leader in this field in personal omics and their applications to individuals and to health. So that was a great opening to the session. The second speaker was Tim Hughes from Toronto, locally here, who's really an expert in splicing and RNA biology. And RNA biology is kind of the next layer, the transcriptome, the RNA is the next layer beyond genomic variation, which modulates how the genome affects proteins and translating into proteins, and how it affects protein function and cell biology. So Tim talked about, in fact, DNA binding proteins and RNA binding proteins, and how we really know very little about many of the proteins that interact with the DNA and the RNA, which subsequently regulate the amount of RNA and the RNA function in the isoforms of RNA and the functions of proteins downstream. And he gave a great insight into some of the new research that's going on in that area. And then we finished by really focusing down on an area of the genome related to human heart disease. 9p21 is related to coronary artery disease and atherosclerosis and myocardial infarction, and it's the strongest locus in the human genome for heart disease of this type. And Daniel Tübzer from University of Munich gave a very elegant talk about a long non-coding RNA, a link RNA, they're sometimes called, a long non-coding RNA, called Untitled, which is at the 9p21 locus. And he presented some strong data to suggest that that non-coding RNA family, Untitled family, modulates modulates many genes in the genome that affect heart disease, and it provided some functional data to suggest particular types of binding elements in Untitled interacting with RNA and DNA. So that was a great session. I think it really taught the audience some new things, as well as being very disease specific. So I enjoyed that a lot. Linking to that, there's been actually a theme throughout the meeting for some very interesting science on microRNAs and their applications potentially as modulators of tissue-specific functions, as well as modulators of therapeutic possibilities. So Catherine Moore's group from NYU talked about a particular microRNA that modulates macrophage function, macrophage autophagy, and there were several talks in the thrombosis sessions on microRNAs that regulated thrombotic pathways. One of the very exciting aspects of the conference are the Saturday morning, or the awardee presentations. Our Hague lecture on atherosclerosis, our keynote speaker, and our distinguished speaker. And this year, from that group, I just want to highlight initially Kathy High from University of Pennsylvania Children's Hospital of Philadelphia, who presents data on a gene therapy for hemophilia. And this is really groundbreaking basic science translating to clinic, where now there's a treatment available, one of the first gene therapies moving towards pediatric disease, the treatment of hemophilia. And Kathy's led this work over the last decades and presented that story. In parallel, interestingly, we had a very complementary story in one of our concurrent sessions led by Marty Ponce, Mortimer Pons, is also at Children's Hospital of Philadelphia, and he presented the use of stem cells to make human stem cells, human-induced pluripotent stem cells, to make megakaryocytes, which are like platelets, pre-platelets, and the use of these stem cell platelets in the treatment of hemophilia for people who would not be responsive to gene therapies or other therapies. So cell therapy and gene therapy for hemophilia and basic science translating to clinic, a very, very important theme in our thrombosis section here. So I would also like to emphasize and highlight the applicants, the strong talks of the applicants for the Brinkhouse Young Investigator Award, and in particular a presentation from Feng Ye from UCSD, University of California, San Diego, on the inside-out signaling of the 2B3A integrin receptor and its interactions with talum, which is a very important functional aspect of how platelets signal inside-out in thrombosis. So switching over to atherosclerosis, which was very strong and interesting at the meeting also, the first theme I'd like to pick up on, there was several talks throughout the meeting that discussed the functional translation or the mechanistic translation of genome-wide association hits or findings in these genome-wide association studies for coronary disease and cardiovascular traits. So now the stage of research for many of those is to understand the function of the loci and the gene that are involved and how they affect atherosclerosis and myocardial infarction and other traits. And in particular, I highlight Daniel Tübster's talk, which was part of the genomics plenary. Claudia Ghosh from Brigham Women's Hospital, who was talking about the SORT1 locus. And several other talks that really were diving deep into the functional translation of these genetic association studies, so that the opportunities for understanding how these loci and genes work in human disease and the opportunities for therapeutic interventions are now becoming more apparent in this area. For the atherosclerosis side, also I'd like to highlight the Young Investigator Award and the Page Award applicants, where there was some very strong talks, and in particular there was two talks that I thought were particularly strong. Babak Razani from the University of Washington spoke about the role of P32 in modulating autophagy and inclusion bodies and macrophages, which is probably an important mechanism of regulating macrophage function in atherosclerosis, and he gave a very powerful and elegant talk, which I think shows how he's come on as a young investigator. So last, I'd like to highlight the vascular biology and vascular medicine front of the conference, which was also very interesting and very strong. I emphasized two or three aspects. I thought Dwight Towler did an outstanding job in talking about the role of calcification in smooth muscle cell and vascular biology and vascular disease in his plenary session. In addition, AJ Chawla gave just an intriguing talk on the role of macrophages in modulating adipocyte function and modulating thermoregulation and how that affects complex traits like metabolism and atherosclerosis. So I'd also like to highlight the talk from Luisa Arispa from UCLA on the molecular mechanisms regulating endothelial cell function in vascular biology, a very powerful piece of molecular work with translational implications. In addition, for the Young Investigator Award again, for the Page Award, Jordan Miller from Mayo Clinic gave a tour de force presentation on molecular mechanisms of aortic valve calcification and how aging and vascular stress may lead to changes in histone through sirtuin-6 in the causation of aortic valve calcification, which is a very important disease of our aging population. And he points for the first time towards some mechanisms which perhaps have some therapeutic implications in the future. But overall, I'd like to summarize by saying we had some outstanding science from senior investigators and from our young investigator invited abstract speakers. I think that poster sessions again were tremendous highlights, where at each of the sessions the rooms were packed and the sessions went well over the time, and there was a lot of networking and scientific and social discussions as part of those. So I think they continue to be a major highlight of our spring conference. With that, I thank you, and I say goodbye from ATVB Conference 2014 in Toronto, Canada.